Molweni Sanbonani Dumilang Salam Habari. Esteemed guests, current and future researchers, and all those joining us both in person and online across the world. I am Yanelisa Pulani, and with me here today is Wahid Amanji. It is an honor for us both to stand before you today representing the African Research Society. When we first began at the University of Cape Town just a year ago, we were a small group of students with a shared dream to elevate Africa through research.
At its heart, African Research Society is much more than just an organization. It's about shaping a community that values African-centric innovation and collaboration, and a community that strives to make a meaningful impact on our continent and the world. Whilst our roots are in the Faculty of Health Sciences, our vision goes far beyond. We see a future where every discipline and every voice has a role in shaping research that is for Africans by Africans. At ARS, we are currently working on bringing together students from diverse fields, ranging from science, health sciences, humanities, commerce, and engineering, and inspire them to develop interdisciplinary solutions tailor-made for our continent.
From hosting research workshops to facilitating networking events, our aim has always been to bridge the gap between students and the world of research. This year alone, we've managed to pioneer several initiatives. The Interdisciplinary Research Apprenticeship Program, IREP for short, is one such initiative that aims to provide undergraduate students with unparalleled research opportunities. Our summer school camp, which hosted preclinical medical students, not only introduced them to the intricacies of lab work, but sought to kindle a passion that will transform them into future clinician scientists, bridging the gap between patient care and cutting-edge research. Our commitment goes beyond the academic labs and lecture theaters. We've joined hands with the science in our schools and are currently working on outreach national benchmark test workshops, which aim to democratize access to higher education by coaching matric students from underprivileged backgrounds before they sit their NPT university entry exams.
We are really ecstatic to see ARS expand its wings with new chapters at universities across the country. It's also really heartening to see all these tireless nights of work bearing fruit beyond South Africa. From the dunes of Namibia to the island of Cape Verde, as well as Eswatini, Malawi, and Mozambique, this journey of growth has taught us that together we can achieve so much more than we could have ever imagined.
Before we move on, a word of thanks is due. Professors Valerie Mizrahi, Salome Maswime, Tobeko Ntusi, Colette Dandara, and Lionel Green Thompson, your belief in our vision has been our guiding star. Today would not be possible without you. And to our honors for the evening, Ingaba Biotech, UCT Department of Surgery, UCT Department of Medicine, Bo Constantia, Clareb Accessories, WizMed, the Neuroscience Institute, and the Institute of Infectious Diseases and Molecular Medicine. Your support has been invaluable. Siabulela, we are truly grateful.
As we look forward to tonight, especially hearing from our esteemed Nobel Laureate speakers, we hope to continue inspiring and being inspired. Our goal has always been simple: to build bridges, to foster connections, and to create a space where every researcher, current and aspiring, feels at home. Thank you so much for joining us on this journey and for scanning in your boarding passes today and for believing in us and for helping us strive towards a brighter future for research in Africa.
So as we prepare to welcome our next esteemed guest, let us offer a glimpse into her exemplary journey. Prof. Salome Maswime is not only the head of global surgery at UCT, but also an accomplished obstetrician and gynecologist. Her passion for leadership and transformation extends as she chairs the University of the Future project at UCT. Her commitment to healthcare and academic excellence is evident through her roles as the chairperson of the Health Systems Trust Board and the founder of the South African Clinician Scientists Society.
Before we continue, I must just note that we probably would have needed a couple more projector slides to fit everything. So please forgive us. We tried to fit as much as we could. Prof. Masume's dedication to maternal health and for pioneering global surgery in our region has not only earned her national recognition, like the Trailblazer Award by the South African president, and most recently, she was the recipient of the South African Medical Research Council's Clinician Scientist Award. And I hear that that event's pretty much like the Science Oscars of South Africa. So yeah. So in the realm of medical research and leadership, Prof. Masume stands as a beacon, continuously pushing the boundaries and shaping the future of healthcare in Africa. Now, please join me in warm welcoming a trailblazer, Prof. Salome Masume, to the stage for our opening note.
Good evening. And firstly, let me just thank the leadership of the African Research Society. It's been such a wonderful journey watching this society doing great exploits on the campus. And I think you guys just don't fail to surprise us. You think big and you do big things. And I think just from the launch in a small, up there in medical school with a very small group, and seeing you guys really just, going further and further, bringing Nobel Prize winners to UCT. I just really want to congratulate you, and I want to say thank you for inspiring us and creating these opportunities. I want to acknowledge Professor Dyer, the Interim Vice Chancellor. I'll acknowledge my Dean, Professor Lionel Green Thompson, and all the leaders in the room. Then I'll say all protocol observed. Today is a celebration. We are celebrating excellence. We're celebrating science. And I want to welcome all of you on behalf of the African Research Society. And I also want to warmly welcome the Nobel Prize laureates, Dr. Charles Rice and Prof. Randy Schekman, who will be joining us shortly and who will be sharing their personal journey with us as well as their academic journey. And in line with the theme of the day, Reach for the Stars, we are about to go into a realm of discovery, excellence, perseverance, and perhaps courage as we learn from scientists who have changed the course of history in physiology and medicine. The celebration of science and the celebration of scientists forces us to reflect on the advances that have been made by those who came before us. It shows us how a person or a group can advance a cause and can advance science and change the course of history with what may start as an idea, an intervention, or an experiment, and how one idea refined over many years can eventually change the world and change how we practice. But it also reminds us that there are still many questions that need to be answered, and we have a responsibility to recognize the challenges of our times and to come up with new solutions and new answers. So hoping that tonight you'll all be inspired, challenged, and encouraged as we reach for the stars and as we learn how to reach for the stars in our disciplines. We live in a continent where 90% of the population doesn't have access to safe and timely surgery, despite the advances in medicine and sciences. So we have a lot of solutions to find to improve healthcare for all in Africa. I love the quote. The electric light did not come from the continuous improvement of the candle. At times, we need to come up with new ideas, new solutions, new answers, or new knowledge to change the course of histories. Scientists see opportunities where the rest of us see a challenge, and they have the ability to bring new ideas and solutions that have the potential to change practice and policy, and sometimes the world. There are so many unanswered questions in medicine, diseases that we haven't fully understood, treatments that we haven't yet discovered. I personally ran a stillbirths clinic a few years ago, and my frustration was, despite all the knowledge that we had, I often couldn't tell patients the cause of stillbirth because many of them were unexplained. So what is your challenge? What questions do you have? What problems do you want to solve? What is your curiosity tonight? It's a great honor for us to meet the Nobel laureates whose research has changed the world, has changed our understanding of the human body, diseases, and has saved lives. Our vision for the University of the Future project is to create an enabling environment to unleash human potential for a fair and just society. With a mission to create spaces, places, services, and functions for the future of UCT. So with that hat, we fully support tonight's mission to learn from internationally acclaimed researchers on how you too can reach for the stars. My hope tonight is that we'll all be inspired and motivated. In nations where young people are exposed to great scientists and leaders early in life, they start to dream and to see what they can do and what they can become. I often wish our country could celebrate science and scientists more to inspire the next generation of African scientists and a generation of African Nobel laureates. So by organizing this event, I say to ARS, you have given us permission to dream dreams that will unleash potential, dreams to improve healthcare, dreams to build a better, healthier and stronger South Africa and Africa. Enjoy the evening.
Hello? Oh, cool. Thank you so much for those wonderful words, Prof. Maswime. Scintillating as always. While we ponder on Prof. Maswime's words for a little bit, we're just going to sort out some technical issues before we get back to the program.
Okay, test 1, 2. Maybe we got the settings here and see. Looks like yes. Settings. 1, 2, 1. Yeah. Let's just turn off the automatic here. Like these are the only things we can change, I think.
I think having that devices might be.
Well, that sounded fine though. It only came through now. I heard it just now because there's quite a lot of delay. Yeah. Yeah, sure. Okay, test 1, 2. We're gonna.
Sorry, guys, the people online just had some technical issues. We'll be resuming the program shortly. Thank you for your patience. Okay, hi everybody, if I can have your attention please. So my name is Sayuran Pillai, I'm a second year medical student, a head of portfolio with the African Research Society. And it is my distinct privilege to introduce to you today Professor Charles Moon Rice, an exceptional virologist whose pioneering research and unwavering dedication has fundamentally transformed our understanding of infectious diseases. With a remarkable journey spanning from his early academic pursuits to his pivotal role in the discovery of the hepatitis C virus, Professor Rice's groundbreaking contributions have not only shaped the landscape of virology, but have also directly impacted global health. His distinguished positions, extensive editorial roles, and many accolades, including the prestigious Nobel in Physiology or Medicine, stands as a testament to his profound impact on scientific knowledge and his enduring commitment to advancing the frontiers of medical research. If you could all please join me in welcoming the trailblazing Professor Charles Rice to our distinguished gathering.
Well, can you hear me? Yes? Okay,
Yes,
well, Sayuran, thank you very much. And, for that very nice introduction. I guess I'm supposed to say a few words, but I'd like to just start by sort of thanking the sort of group of enthusiastic young researchers for the opportunity to, spend some time with you today. And I thought in my remarks, I'd just sort of give you a little bit of a history of my career, which I don't think is really as sort of special as is sometimes touted, but actually sort of relatively simple. And I think when I finished my undergraduate degree, I wasn't really quite sure what I wanted to do, but I did apply to a few PhD programs and went to Caltech. And at that time, I was, thinking of being a developmental biologist, but I ended up in a virology lab, which I didn't expect, and I really enjoyed, sort of working on viruses, and that really sort of became my career. But it was really very much that sort of chance placement when I arrived at graduate school that, sort of gave rise to sort of my career. Now, in terms of a precursor to our work on hepatitis C, we sort of go back to really something that, sort of originated in Africa, and that is yellow fever, which I think many of you know. And as a sort of a late stage graduate student and sort of postdoc, we became interested in yellow fever and this particular group of viruses called Group B arboviruses transmitted by mosquitoes, and started working on yellow fever, the vaccine strain that Max Tyler basically produced in the mid-1930s, which has been in about 500 million people worldwide. So really quite a remarkable accomplishment, which Max won the Nobel Prize for in 1951. So yellow fever, I think, was an interesting member of this group of viruses to work on because you could actually work on the vaccine strain, so it was relatively safe to do. And I think that the sort of the pivotal point in my career was when I was an assistant professor at Washington University in St. Louis, and working hard on yellow fever still. And then in 1989, this paper in Science came out from Michael Houghton and Harvey Alter on non-A, non-B hepatitis, identifying a new virus which they called hepatitis C virus, as the cause of not an arboviral disease, but actually a disease that was noticed by Harvey in transfusion medicine. And so it turned out that this new virus was very similar to yellow fever. And so as a young assistant professor, I had to decide if I was going to work on that. And that was actually catalyzed to some extent by a call from Steve Feinstein, who was at the FDA in Washington, interested in a paper that we just published on yellow fever, reporting a genetic system that allowed us to make engineered changes in the viral genome. And he was interested in making a vaccine for hepatitis C. And so that was really the sort of push that got us working on that virus actively. And the problem with hepatitis C was that the virus didn't grow in cell culture. So it's not like, SARS-CoV-2, where you can take a swab from a person who's showing symptoms and basically have a virus stock in a couple days in the laboratory. That wasn't the case for hepatitis C. And really a series of efforts to sort of develop systems that would allow us to study this virus and develop new treatments sort of occupied the next 15 or so sort of years of research, starting in 1989, sort of the discovery of a missing piece of the viral genome, which we made in 2004 and 1995. Then finally making a functional molecular clone of the virus, which we had to demonstrate in the chimpanzee model in 1997. That still didn't give rise to a system that allowed the virus to replicate in cell culture until 1999, when Ralph Bartenschlager and Volker Lohmann reported the replicon system as a truncated version of the viral genome that could replicate in a human liver cancer cell line. And then even that didn't give rise to a system that allowed the full virus life cycle until 2005. So really each one of these breakthroughs was punctuated by another roadblock, frustrating for a molecular virologist like me. And, I think it was, if you have to think about what was, an important lesson that was learned by all of us sort of working on this problem, was that, persistence really does, sort of pay off. And the discovery of hepatitis C led to the realization initially that there were over 100 million people that were chronically infected with this virus, and we didn't have a good treatment. And so there was a huge unmet medical need to clean up the blood supply, since this was a blood-transmitted virus, not an insect-transmitted virus, and develop new treatments. And then we encountered all of these technical hurdles that we had to go through in order to make that a reality. So from the discovery in 1989 to sort of these molecular tools that came in the late 90s and early 2000s, to the complete virus life cycle 15 years after the virus had been sort of discovered, did catalyze work by many, many hundreds and thousands of people around the world trying to understand more about this virus, and also to develop inhibitory compounds. And really the first of those compounds started getting used in 2011 in combination with interferon therapy, which was basically the only treatment that worked for hepatitis C, but had terrible side effects that many people couldn't tolerate. And then in 2014 was really the advent of being able to treat people with a cocktail of drugs and eliminate the virus in almost everybody that was treated. So we now have treatment regimens that basically can cure 100% of the people treated, either with one course or with two courses. Usually one course is sufficient to cure more than 95% of the people who are treated. So, for a fundamental investigator and a researcher to be able to be involved in the relatively early stages of this quest to actually being able to see sort of patients treated after a month or two of treatment with few or no side effects is really, sort of a special, sort of experience and honor for me to have been participated in. And I would also say that, this is an example where, receiving something like the Nobel Prize, at least, was not something that I ever sort of entertained. I mean, it was really just the curiosity about this virus and really believing that, we could make steps forwards by, research in the lab, and that would, sort of help to sort of get to the finish line. And really, these awards are given to just a few people, but there are, hundreds of people that were involved in this work in my laboratory over the years, and also around the world with many fantastic colleagues, both in academia and industry, and doctors in the clinic and patients participating in clinical trials that have gotten us to where we are today. And I would just say to all of you out there in the audience that, the tools that you have today for doing research and understanding, sort of fundamental biology are really just mind-blowing. It's really going to be the next years in research are going to be like no others. And I think you have a very bright future ahead in solving some of the real problems that we face in the world today. So I don't want to go on for sort of too long. I know that the program has to continue, but I'm very much looking forward to sort of chatting with you sort of later in the evening and hearing about, sort of what questions you might have. So thanks. And I guess we'll move on to the next step in the program.
Good evening, everyone. My name is Titme van Heerden, and I'm the African Research Society Secretary General. I am an MBCHB student, currently doing my honors in neuroscience and physiology as part of the intercalated program. And I have the honor of introducing our second Nobel laureate for the event, and that is Dr. Randy Schekman. Dr. Randy Schekman is a Nobel Prize laureate whose groundbreaking research alongside James Rothman and Thomas Südhof unveiled the intricate machinery orchestrating cellular vesicle traffic, a fundamental process that underpins life itself. As a revered professor at the University of California, Berkeley, Dr. Schekman's pioneering insights into cellular transport have illuminated the path to understanding health and disease. Beyond his lab, his editorial leadership at the proceedings of the National Academy of Sciences and his profound influence on budding scientists all over the world underscore his far-reaching impact. Born on December 30th, 1948 in St. Paul, Minnesota, his journey from a UCLA molecular sciences degree to a PhD in biochemistry from Stanford University embodies an unwavering pursuit of scientific excellence, leaving an indelible legacy inspiring many to the quest of knowledge. I must say it is unfortunate that we were unable to have Dr. Randy Schekman join us in real time today due to unforeseen circumstances. However, we were lucky enough to have conducted and pre-recorded his talk as well as his interview, which we will be playing now. He has also very kindly agreed for us to set up a post-event Google form for you to submit your questions, where he will kindly answer some of them when he has time. But without further ado, I present Dr. Randy Schekman.
My name is Randy Schekman, and I'm a professor at the University of California, Berkeley, and an investigator of the Howard Hughes Medical Institute. It's my pleasure to be able to address you today. I'm sorry I can't be with you, but I hope this recording will be a substitute. I'll start by giving you a very brief introduction to the work that I've done over the past now 47 years here at the University of California, Berkeley. I began my career in graduate school studying the mechanism of DNA replication in the laboratory of one of the 20th century's great biochemists, Arthur Kornberg. And from him, I learned how to take a complicated problem apart piece by piece using in that laboratory using primarily biochemistry. I decided to switch fields for my postdoctoral work, and when I began at Berkeley, I wanted to explore the mechanism of protein secretion. This subject had long been established as the domain of cell biologists who study animals and use either cultured animal cells or laboratory animals and tissues to study the mechanism of secretion, like molecules such as insulin, primarily in exocrine tissues such as the pancreas. But in the mid-1970s, studies on a simple eukaryotic microorganism, Baker's yeast, became very popular because of the tools available to dissect complex cellular processes using yeast. And the reasons for this are quite simple to explain. Yeast cells are fundamentally very similar to the cells in our body. And here is a cartoon that I use to illustrate this point. On the left is a painting of Charles Darwin, and on the right is a sample of such as you might see growing on the surface of a grape. Outwardly, of course, these two don't look very similar at all, but yeast cells have much of the basic machinery to operate inside the cell that almost all of the cells in our body have. It's easy to study yeast because you can grow it in large quantities. You can produce a uniform population of billions of cells, and much is known about the genetics of yeast, even when I started nearly 50 years ago. Here's a high magnification view of a yeast cell showing the bud portion of the cell, which has grown to approximately the size of the mother cell, at which point the cell puts down a septum and the two cells separate. Here is an earlier step where the bud has just emerged, and it will continue to grow to approximate the size of the mother cell over the next 90 minutes or so. When I began my laboratory, it was with the belief that this part of the cell surface, in this case you're looking at a cell wall, would expand during the growth of a yeast cell, during the cell division cycle, by the insertion of new membrane material into the bud portion, the tip of the cell. And by a process of protein secretion, the bud would grow and enlarge until the time of cell division. This is more evident in looking at a different electron microscope view. This is a thin section of a yeast cell. It looks very different than a mammalian cell, but it has many of the organelles that were identified in mammalian cells associated with secretion. There's a structure that is similar to the lysosome called the vacuole. There are tubules of membranes that represent the endoplasmic reticulum and can be seen in other sections to have ribosomes bound on the cytoplasmic surface of the membrane. In other sections through the cell, you can see a nucleus that is an extension, whose membrane is an extension of the endoplasmic reticulum, and you can see elements of the Golgi apparatus. But what struck me and one of my first graduate students, Peter Novick, was a collection of small vesicles that congregate under the bud portion of the dividing cell. Now, these vesicles had been seen before by work prior to the start of my lab, and it had been shown that these vesicles carry enzymes that get secreted into the cell wall that surrounds the yeast cell. And so we imagined that these vesicles, which have a bilayer surrounding this inner substance of secreted proteins, would become the building block for the growth of the plasma membrane. So we imagined that by a process of membrane fusion, new membrane material would be added to the bud and would grow step by step during the 90 minutes required to form a new bud. So here's Peter Novick shown at his bench back in 1978, just after we began the work. He had very long hair back then, like we all did. He's now practically bald, as I am as well. And so, but we've aged well, and he is now a very prominent cell biologist on his own right in his laboratory at the University of California, San Diego. So we began to search using genetics for mutations that block this process. We imagined that if you crippled any of the genes that would be required for the production, for the movement, and for the fusion of a vesicle at the bud plasma membrane, such mutations would kill the cell. And so we were looking for essential genes. Of course, most of the genes in yeast cells are not essential for cell growth. Maybe 30% of them are essential. And the way you look for such genes is to look for mutations by random mutagenesis of yeast cells, mutations that change amino acids that render the protein thermally unstable. Yeast cells can grow at a range of temperatures, from room temperature up to near 40 degrees. But if you have a mutation that changes an amino acid that is structurally important for the stability of the protein often will function normally at room temperature, but fail to function when the cell is raised to a higher temperature, such as 37 degrees. So we began with a very simple quest to look among random temperature-sensitive colonies that were produced by random mutagenesis for those that caused two different enzymes normally secreted into the cell wall to be accumulated inside of the cell when the cells were incubated at 37 degrees centigrade as they were in the process of dying at that temperature. We found the first such mutant called SEC1, and with encouragement from the most prominent cell biologist studying membranes at the time, George Palade, we examined this cell, the mutant cell, after incubation for a couple of hours, 37 degrees, by thin section electron microscopy. And in contrast to the image you saw a moment ago, these cells, SEC1 mutant cells, accumulate thousands of vesicles spilling over and filling the entire cytoplasmic volume. No longer restricted to the bud, these vesicles appear to fail to be delivered or diffused to the plasma membrane, and they lose their way and they spread out throughout the cytoplasm. Importantly, when these cells, having accumulated vesicles, but before they die, when these cells are returned to room temperature, permissive growth temperature, the vesicles re-engage the secretory pathway, their content is secreted and the cell can resume growth. So these are temperature-sensitive, thermally reversible mutations. Well, this was very exciting, and it launched a vigorous effort over the next year and a half to isolate many more such mutations. After efforts by Peter Novick and my technician, Charles Field, we identified an additional over 200 different mutations, and were able to define a genetic map of the secretory pathway. After much further work, we could show that dozens of such genes could be identified, and on close inspection, both by thin section electron microscopy and by the creation of double mutant cells, we could establish an order of events. We found mutations that block the very first step, the insertion of proteins through a channel in the ER membrane. We found mutations that block the bidirectional flow of small vesicles between the ER and the Golgi apparatus. Subsequent postdoctoral fellows for my lab, Scott Emmer, and others were able to isolate mutations. Tom Stevens were able to isolate mutations that define a limb of the pathway leading from the Golgi apparatus to the vacuole. And finally, mutations that block the very last step, including this gene, SEC1. We now know, subsequent to our work, that these genes can be found in the human genome. SEC1, for instance, encodes a protein required in almost all cells, and particularly in the brain, it's required for the fusion of synaptic vesicles at the presynaptic membrane. And the same is true for all of these genes, affirming the conviction that I started this with the picture of Charles Darwin and yeast cells. There is a fundamental evolutionary connection. Well, in addition to providing an opportunity to discover the functions of these gene proteins, which I don't have time to tell you about, since we were able to show that this pathway is so fundamentally conserved, the biotechnology industry, which was growing up in the San Francisco Bay Area, decided to use yeast as a platform for the manufacture of commercially useful quantities of clinically important proteins, such as human insulin or the hepatitis B vaccine. And now, many years later, 1/3 of the world's supply of human recombinant insulin is made by yeast cells, which have been where the human insulin gene has been introduced and where it's expressed in abundance. And now you can isolate.
Okay, so this next part of the evening will be an interview, and the interview will be in two stages. The first part will be the pre-recorded interview that Sierra and I conducted with Prof. Randy Schekman. We hope you enjoy the pre-recorded interview, and then after that we will be conducting an interview with Prof. Rice.
We heard a lot about your research, but I was wondering if you could actually tell us a little bit more about your actual journey into the field of cell biology and how you found yourself wandering into the world of science and research in general.
Well, happy to do that. That, of course, takes me way back to when I was growing up in Southern California. We lived in what is Orange County, which is now a very heavily populated part of the LA basin south of the city of Los Angeles. But at the time, it was still very suburban, somewhat rural. And near my home, there was a still creek with some pond water with scum on the top of it. And I remember, for reasons I can't reproduce, wandering down there one day to collect a jar of dirty water with the film of pond scum. And I had at this time I was maybe 12 years old, I had a toy microscope that I'd gotten for a birthday, I think. And I put a drop of this on a glass slide and looked at this world that was real, even in the toy microscope, that I couldn't believe. These organisms swimming around and crawling and bumping into each other. It was a chaos and a diversity that you don't even see with the naked eye looking out to the world. It was quite remarkable. And I was just taken with this, and I spent hours looking at samples and seeing the diversity of life. And one day at dinner, I was excited and expressing what I'd seen in the microscope, and my father was rather skeptical, since this was, after all, just a toy. And I remember being somewhat offended at his skepticism. And so I resolved at that time I was going to save money from the many chores that I did to buy a student professional microscope. And so I mowed lawns and babysat for neighbors' kids and delivered newspapers, and I kept the money in an envelope in the closet in my bedroom, but my mother kept borrowing the money, so I didn't actually get to the $100 that I needed. So one Saturday morning, I remember vividly, I was mowing a neighbor's lawn, and I was so upset that I rode my bicycle to the local police station, and I told the duty officer that I was running away from home because my mother was stealing my money and I couldn't get my microscope. So they called my father in, and the captain spoke to him behind a closed door, and he came out looking rather severe. And but I learned a valuable lesson that afternoon, because we then drove to a pawn shop in a nearby city, and I purchased for the $100 a Bosch & Lomb student professional microscope, which became my pride and joy for the rest of my time at home and through school. And of course, that microscope got put away when I went away to university because they had better microscopes. But fortunately, my parents saved that. And when I then later in life had a job and moved to Berkeley, they sent it up to me. Unfortunately, neither of my children had any interest in science. So the microscope sat around collecting dust in a closet until one day, after the Nobel Prize was announced, now 10 years ago, I got an email from the Nobel Museum in Stockholm. Every year they ask laureates to present some artifact that is a reflection of their discoveries, so that it could be on display for others to see. And I thought, what better could I do than to send that microscope? So if you ever get to Stockholm, you can visit the museum, where you will see my microscope on display, together with a caption in English and in Swedish, indicating that I had to run away from home to begin my career in science.
Yeah, wow, what a wonderful story. And really seems like cell biology was a passion for you right from the beginning. That's so amazing.
Well, microbiology was really
microbiology.
And, throughout college and graduate school, I studied bacteria and their viruses. And when I started my own career here, it was again with microbes, in this case with eukaryotic microorganism.
Perfect. So Prof. Schekman, you told us a bit about your childhood with your parents. So your father was an electrical engineer and computer software designer, right? So
Yes.
How do you believe that his background might have influenced your own journey in the field of science and perhaps shaped your own perspective on research and innovation?
Well, he was an engineer, really, and so he was good at mathematics, and so I learned that appreciation from him. But he also tried to get me interested. So he was actually very early in software design, even in the 1960s. And one summer, when I was after my first year in university, he got me a job in his firm, where he hoped that I would become interested in coding, which was, of course, very cumbersome back then. And I actually found it really boring. So I spent the summer thinking about what I might do in a research lab, again, working on microorganisms. So although he may have stimulated my interest in mathematics, he was not responsible for my interest in biology.
It's so wonderful that you really had your own journey, and that you discovered something sort of by yourself. You discovered this passion through your own experience, which is wonderful. And as we know, you were awarded the Nobel Prize, as you said, 10 years ago now. along with James Rothman and Thomas Sipple. And we saw that you were all looking at sort of slightly different pieces of the same puzzle that eventually came together. And obviously, I imagine this sort of involved quite a bit of teamwork. And so we were wondering, what has your experience been like working in a team throughout your research career?
Well, that's a great question. Rothman and I were more competitors than teammates. we had the same overall goal, but very different approach. But we did, and so we were very competitive, and it was quite tense in the early years, trying to outdo one another. But at a certain point, when some of the genes that we had discovered turn out to encode the yeast equivalent of the mammalian proteins that he had purified using biochemical approaches, there was a kind of a golden spike that put the two approaches together, and we became more allies than competitors. So in fact, we're now good friends, and those bygone years of competition are long past. But you raise a very good point that I feel very strongly about. Somewhat later, after that period where we had more closely aligned, I became involved in a long-term collaboration with a very accomplished electron microscopist, an Italian scientist working in Geneva, who had already collaborated with Rothman for his work. And so I was delighted to be able to collaborate really intimately with this Italian Swiss, who had skills that were beyond mine in morphologic inspection, but where he had no knowledge of genetics or biochemistry. And that collaboration, which lasted for 15 years, was really remarkable. We spoke virtually every day. We shared samples and results, and I made the pilgrimage to Geneva many times. And that was magical. And I've learned since that experience how important, really meaningful collaboration can be. And I'm now involved in a very large-scale international effort to discover the mechanism of Parkinson's disease. I don't know if you wanted to get into that later in our conversation, but this is now an all-consuming effort of mine, and it involves 163 different laboratories organized into 35 teams in 80 different institutions, and it's all of the investigators are linked in a virtual collaborative research network, where the goal is to bring all these people together in really meaningful, real-time interactions to push the needle forward on this intractable disease. So I actually have become full circle, although my first years as an independent investigator I was flying solo. I now really believe in the value of collaboration.
That's brilliant that you could have that full circle moment. And I think it must be so amazing to be part of such a huge collaboration. And I think it just goes to show how far we've come in the world of research and science, and that how much more willing we are to actually work as a team and to say it doesn't, always have to be competitive. Like healthy competition is brilliant, but teamwork is so valuable.
Well, yeah, there's one factor works against that, and that is unfortunately the reward structure for academic scientists favors the individual. It doesn't really favor teamwork as much as teamwork is favored in enterprise, in businesses, or certainly in biotech. So there are problems with the academic style, the academic approach, because the reward structure typically is given not necessarily in money, but it's given in authorship on papers in these so-called high-profile journals. And that does not necessarily encourage meaningful collaboration, because people end up fighting over where their name is placed on a list of authors, and that is antithetical to really meaningful collaboration. So that's something that we have to get around. So
you spent the next 20 or so years committed to this research, and it seems like a really large undertaking, as I'm sure it was. So as you navigated these various stages of your career, how did you manage to strike a balance between, your personal pursuits and your professional life? And what advice would you offer to young scientists to maintain such a balance?
Well, there's a lot of discussion about work-life balance now that it wasn't true when I was a student. When I was a student, I worked day and night. I spent many full nights in the lab. I was completely obsessed. And when I started here at Berkeley, I was married at that point, and my wife and I wanted to raise a family, so we had 2 children. And I was raised by very attentive parents, and I felt that it was important for me to be there for my kids and not to be continue to be completely obsessed. So I think my kids knew me growing up. I would be there evenings and weekends to be with them. I think what you have to do to have a balanced life is to be better organized than you might otherwise be. You know, some people who work constantly, day and night, are not necessarily very efficient. You know, people spend a lot of time just in idle chat and not doing things that are not directly on point. And so if you want to have a balanced life, you have to really focus and do what's needed during the time that you wish to devote to your professional activities. So, I mean, I certainly, took some work home, but I wasn't terribly efficient at doing it at home. I was more efficient doing it in my office. And people who remember me from back then saw me just scurrying about, focused on the task at hand. And I think people need to be better organized if they want to have a balanced life.
That's a really great tip. I think we're all writing it down.
Making notes.
Well, it's easier said than done, but you have to, you have to put your, establish what your priorities are and work on those and not be deflected by other things.
Yeah, definitely. So we're sort of coming to the last question, and probably for us the most important question. So looking back on your life and your career and this magical discovery, what piece of advice would you offer to your younger self as you were just embarking on this journey into the world of cell biology and research?
Mm-hmm. Well, I often think about this, and when I was in university, and particularly when I was in graduate school, I think I was very insecure. I went to university at a public institution at UCLA, and it was a fine institution. It gave me all the tools that I needed to succeed. But the department where I was a graduate student was at Stanford, and it was at the time the most selective, most prestigious place to do nucleic acid research. I think certainly in the States, possibly in the world. And so most of the graduate students who came to that department were, from very selective private institutions like Yale and Harvard, MIT. And I felt insecure at first about that. And so, unfortunately, my behavior was kind of obnoxious, I have to say. I was compensating, overly compensating. And as a result, I was personally isolated because of my attitude and behavior. And so if I were able to communicate with myself now 55 years ago, I would say, close your mouth and be polite. Talk about other people and not be boastful and listen rather than just speaking. Now that was me. Other people maybe were better behaved when they're younger, but I was not. And I suffered because I was isolated in that experience. And so subsequently, as my personality evolved and my work advanced, I think my better self now is what operates, and I'm much more friendly and open to new people than I used to be. So that was for me. I don't know that applies to anybody else, but I think that was really a failure on my part when I was much younger, and I would have had a much easier time of things if I had been more thoughtful and respectful of other people.
That's a really powerful sentiment to close our discussion on.
Yes, and I think even the point you made about insecurity, even if you might not face the other challenges, I think all of us face insecurity in some aspect of our lives. And maybe having a bit more self-belief will open you up to more engagement and more collaboration.
One of the things that came from my graduate career, in addition to realizing that I had to be respectful of other people, was as my experiments worked and the work developed very well, I developed a confidence, and not arrogance, but a confidence in my own abilities that I may not have had because of my insecurity. And that confidence is what got me through those difficult times, like when my grant was trashed. Because I knew that my ideas had merit and had been proven in other contexts, and I was ultimately certain that I would achieve what I wanted to do in spite of the reservations that others may have had. And it was only that confidence was only as a result of the success that I had as a graduate student. So one of the things that I've learned is for young people who want a career in academic science or, in exploring the unknown, you have to gain experience to develop self-confidence. And then you have to be willing to take risks. If you have that confidence, you have to be willing to take risks, because if you're not willing to take risks, if all you do is you just continue to turn the crank from what you've done before, you will never explore really at the cutting edge. I think many scientists are very risk averse. They do the next obvious thing, and they don't challenge themselves to think beyond that. And I think many of the best, most successful people are really willing to take risks, and of course mitigate the risk by doing, being very thoughtful about the approach. And so another thing that I learned this as a student, all those things work best if you really focus. Some bright people are so bright that they just, they work on all their bright ideas, and then they diffuse their effort, and they can't make progress in one direction. I mean, there are very few Einsteins who can do that. Most people, if they have a good idea, good, focus on it and just stay with it, and challenge yourself to think in new ways about how to solve a really interesting, complex process, and don't be diverted by all the crazy ideas that you have, because most successful people, I think, have succeeded because they've been obsessive about a particular goal and have continued to challenge themselves to approach that goal, to finally get to a level of understanding that they find very satisfying. And anyway, that's been my thought. It's not, there are many different paths to success, many different styles. I don't say this is the only style, but it really worked for me. And the people that I see who've been most successful have been that way. They really had picked a big question, a challenging question, and they've been obsessive in pursuing that goal.
Yeah. Well, thank you so much for those really brilliant words of advice. I think we really appreciate it, and I know I'll
Okay, so I hope you enjoyed that recording. The last sentence was, we, the audience, would appreciate the advice as well. I'm sure you do. Sarah and I have sort of been sitting awkwardly in the audience. It's quite intimidating seeing your face up there larger than mine.
Hopefully we look a little bit better now than we did. We were younger then.
Yeah. We're so much more mature now. So now we want to start in real time interview with Professor Rice. Can you hear us, Prof. Rice?
Yeah, I can hear you. Can you hear me?
Yeah, wonderful. Yeah. Okay, well, let's get started. So
Yeah, there's a little bit of an echo on the sound on your side, so you may need to repeat the questions a couple times for me to catch them.
Okay, no worries. Just let us know. Okay, so let's begin by delving into your early academic journey. You initially pursued an undergraduate degree in zoology and then later ventured into biochemistry. What motivated this transition, and how did it shape your perspective as a scientist?
Okay, I had a hard time understanding it. Let's do it again.
Okay. I'll try to break down the question to be. There's an echo. Just like the text means about the echo.
Just some technical difficulties again with this university. Thank you.
Okay. Okay, I see what you're saying. Yeah.
Me.
When you ask him the question, he starts talking about Should I just ask now?
Yeah, just type the question in the chat.
Okay, try ask now on the microphone because I've muted the speaker.
Hey guys, we can't go further. Okay, you want to proceed with the event and I'm going to type the questions into the box.
Actually, I can hear you guys at the podium, but that seems to be clearer than sort of via the mic.
Oh, can you hear me now?
Yeah.
Oh, fantastic. Terry, just stand
Is
here.
there an echo now? Is there no echo?
Yeah, I can hear you now.
Okay, great. Well, can the audience still hear us? Okay, great. Sorry for all the technical difficulties.
Oh,
Okay,
that's okay. You would think after, 5 years of doing Zooms, we would have it down by now, but.
Yeah. Yeah. Okay, so I'm going to do the question again. So I wanted to say, let's begin by delving into your early academic journey and your early research career. So you initially pursued an undergraduate degree in zoology, and then you later ventured into biochemistry and ultimately ended up in virology. What motivated these transitions and how did it shape your perspective as a scientist?
Yeah, well, that's, I guess this is an example of random collisions and random walk in a certain extent. I went to, sort of UC Davis, thinking that maybe I wanted to be a veterinarian. This sort of public sort of university has a very good vet school. And then there I encountered a really fantastic teacher in my introductory biology class, Dennis Barrett. And I really sort of enjoyed that class a lot and ended up actually doing some research on sea urchins. And I think, kind of like what Randy said about, taking a look at pond water, through a microscope, being able to watch, fertilization of, just by sort of mixing eggs and sperm together of sea urchin and watching these sort of transparent activities going on, I thought was kind of got me hooked on biology. But I think as an undergraduate, I was very unfocused. I was taking classes in math and chemistry and biology and zoology, and the sort of zoology degree ended up just being somewhat of I had the right number of classes in that discipline to graduate at the end of my time. And then on to sort of Caltech, where one of the reasons that I went there for my PhD was that I was, sort of interested in sea urchin development after my, sort of undergraduate research. There was a really great lab there. But instead of getting placed in this developmental biology lab studying sea urchins, I got placed in a virology lab. So that was really, just by virtue of, I don't really know sort of why I didn't get assigned to the sea urchin lab, and I got assigned to the virology lab. But I guess it just sort of means that I can get interested in just about anything. So I may be the sort of the example of what Randy said should be avoided, and that is, getting too scattered as opposed to focusing. So anyway, that's a little bit of my trajectory, which is, sort of not the, I want to be a doctor when I was 5 years old and I stuck with it. but really sort of a series of sort of chance encounters that sort of determined my trajectory.
Well, that's really interesting, Dr. Rice. I think it really speaks to how we have to be adaptable and willing to change with new circumstances and willing to make the most of it. So also, interestingly enough, around less than a year ago, last year, I think, we hosted Dr. Harvey, who also worked with you on the hepatitis.
Yeah.
So we wanted to ask, you must have really worked together for a long time. Could you share more about your partnership and about how the spirit of collaboration contributed to your groundbreaking discoveries together?
Well, actually, my connection with Harvey Alter is kind of remote in a way. Harvey, as a transfusion medicine sort of doctor, was really the one, along with Fred Prince, who worked at the New York Blood Center here in New York City, that noticed that people were coming down with this mysterious liver disease that was not due to the known hepatitis viruses at the time, hepatitis B and hepatitis A. And Harvey, as he probably told you sort of last year, had a transfusion medicine patient who'd had some open heart surgery. And this was a famous patient of his called Patient H, or Mr. Hutchison. And Harvey had the foresight to actually collect plasma from Mr. H in the acute phase of his infection from that he got from these transfusions. And this was in 1977, so it was called H77 was that isolate. And that became kind of the preferred HCV, sort of non-A, non-B hepatitis material for the field. And so when we decided to start working on this, I got a little bit of this famous, sort of patient H serum to begin to try and do some, sort of molecular virology with. And that came via my colleague Steve Feinstein, who worked at the NIH and did work with Harvey Alter, and he had some of this material which he sent to us to sort of clone and characterize. So, but sort of since that time, I've gotten to know sort of Harvey very well, along with Michael Houghton and the sort of other co-laureates, and sort of really responsible for these tremendous breakthroughs that actually position us more sort of molecular virologists in a place where we could actually do what we knew how to do. So I think it's the hepatitis C field has really been I think very collaborative, competitive at times, but I think we've respected the advances that our competitors have made if they move the field forward, rather than sort of being upset. So, and people have been very good about exchanging materials and so on. So it's actually been a very rewarding field to work in.
That's so wonderful to hear that it was so rewarding and actually involved quite a bit of collaboration. And actually, I think we wanted to ask a little bit more about teamwork in general.
Yeah, so what has your experience been of working in a team throughout your career? And are there any key lessons that you learned about collaboration that you could share with us?
Well, I think, when you decide that there's a question that you really are curious about answering, I think that persistence, sort of really, and focus really does, pay off. And so I think that sort of desire to, sort of really probe the unknown, and not be discouraged when things don't work out the way that you expect. And actually, I think if you're studying a particular aspect of biology, there's so much that we don't know. It's very difficult for anyone to sort of predict, sort of what the outcome of a particular experiment is going to be. although, some people have certain expectations and they formulated a hypothesis that, they're hoping to shed light on by an experiment. I mean, I think what you often hope for is really sort of an unexpected result, which could mean that you mixed up samples or whatever. So you have to go back and, sort of repeat these and have confidence in your experimental, sort of technique and your results. But it's really those unexpected things which, that may, sort of throw you off track initially that I think lead to, sort of discoveries that, would be unanticipated with what we know now. So I always enjoyed the journey, just sort of getting, even if it's a long journey, sort of solid steps along the road, high quality data that you can really sort of trust. A gel that looks beautiful, simple things are often sort of one of the pleasures of doing research.
That's really interesting to hear about what you really love about the world of research. And as we all know, mentorship is really important in research and crucial in scientific growth and actually important in all aspects of life. And as we know, you had the privilege of learning under the guidance of Professor James Strauss. And we were sort of wondering if there were any specific lessons or principles from him or any other mentorship that you were under that have influenced your own approach to mentoring young scientists.
Well, I think, sort of mentoring is much more appreciated today, in terms of the importance of mentoring. I don't think that, any of the people in my career that sort of turned out to be, sort of fantastic mentors were, had been trained to do that. I just kind of sort of lucked out, with my introductory biology teacher at UC Davis, Dennis Barrett, and then also, sort of in the Strauss lab. But I think that one of the things that I learned is that, it's important to give your trainees, sort of freedom to explore their own ideas, rather than, always being 2 steps ahead and thinking of what they should be doing and telling them exactly what to do. To really sort of let people spread their wings, if you will, even if it's, sort of not the best path forward, because I think sometimes you learn more from things that don't work than you do from things that do work. And so, addressing what appears to be failure and overcoming it is really important. And then letting people sort of have their own thoughts and letting them communicate it. I think in the Strauss lab in particular, and at Caltech, I mean, that was an exciting time, just as we're experienced the sort of revolution in sort of bacterial genetics and bacteriophage interactions that, have given rise to, CRISPR and our ability to sort of use that technology for research and developing new treatments. In the mid to late 1970s, that was the sort of recombinant DNA, revolution. And so, I was given a lot of freedom in the Strauss lab to assimilate some of this new technology and apply it to RNA viruses. And, so it was, I really learned how to sort of move into a new area and really try and apply it to in an aspect of biology where it hadn't been applied yet. So I think that's important today. I mean, I think mentorship is more important. I mean, there's just a lot more going on. There are a lot more people doing science. It's harder to get noticed. And I think, scientists in your generation that are really sort of serious about their careers are, sort of asking for, advice of established scientists who are role models and who have been successful, and trying to sort of learn from those experiences and, not make the same mistakes and get some advice on how to move forward. At the end of the day, you've still got to be, the one that sort of decides what you're going to do. But, sometimes more information is a good thing. And I think also, when you take on a PhD student for training, it's not only sort of completing your, sort of PhD degree, but also making sure that that young scientist, sort of has the confidence and the tools to go into new areas and feel comfortable exploring them. And, very much in terms of what Randy Schekman was saying, that self-confidence that comes from, tackling a tough problem and succeeding is something that, sort of goes on for and needs to be there for the rest of your career. So yes, I think mentorship is important. It certainly doesn't stop as a mentor when your student graduates and finishes their PhD and goes on to a postdoc and, moves into academia or industry. It's important for mentors, I think, to sort of stay engaged and be as helpful as they can in terms of, allowing our trainees to have the careers that they want.
Well, thank you for that, Dr. Rice. I really took away the idea of allowing trainees to explore wrong ideas and allowing them to learn from that. That was really interesting. And you spoke a lot about how you can have a good influence on other people's development as they grow. Were there any pivotal moments in your early stages of your career that impacted your approach to scientific inquiry or guided the trajectory of your career later on that you could tell us about?
Well, I think for me, doing research as an undergraduate, I got to experience that, as is many times the case if you're lucky enough to be at an institution where, that's an option. But, what were really sort of transformative experiences for me, and again, this probably was in part the doing of my sort of biology, sort of introductory biology teacher, was to, go to one of these summer courses where you are, sort of one of a number of students really spending 24/7 doing sort of research and, sort of learning new aspects of biology. And I had the great fortune to take a physiology course in Woods Hole, Massachusetts in 1973 that had, just a fantastic collection of instructors who were, probably using this as an excuse to spend their time out on the Cape during the summer. But at any rate, as students, we could take advantage of their lectures and also, very intensive sort of work in the laboratory to sort of learn new technologies. And I just found that to be really just fantastic, to be surrounded by a number of other sort of young people that thought they might want to be scientists that were sort of experiencing this. After that, I think it was hard for me to go back to, my senior year in college, because I really, just enjoyed, sort of doing research, even though I was still not quite sure at that point. But I think it was the ability to actually see what it was like, for 2 months in the summer, to just sort of devote all of your effort to that. I wouldn't say all of their effort. I mean, we did have, play football and baseball and, probably went drinking at the local pub and things like that. But it was, I think for me, a transformative experience.
So lovely to hear about that. And I think it's quite insightful for the audience and for us to hear that even if you're unsure of something, you can still make such a successful and brilliant career in research and in science, and that sometimes you don't have to be 100% sure, you just have to give it a try. And
yeah, well, in my case, and you may appreciate this in South Africa, I was teetering between science and winemaking. Because I actually had, as part of my very diverse undergraduate education, I had taken a few viticulture and enology courses at UC Davis, which has quite a sort of world-renowned presence in the winemaking industry. And so, yeah, I was teetering on the edge for a while, and so I did get back into that later on, but that's another story.
Oh, wow. Well, perhaps if we have time at the end, we can hear more about that story.
Just wanted to note that I took away that the exposure that you got in your undergrad years was really kind of important in you making those steps forward in research. And I just also wanted to say that that's exactly what the African Research Society is all about. So thank you so much for sharing that with us.
Yes, thank you so much for that sentiment. And then we heard a little bit about Dr. Schekman and the froth on the river and his first microscope and his journey as a young child getting into science. And we're sort of wondering a little bit about your upbringing and your early life experiences, and something that may have influenced your initial interest in science.
Well, I think I was always interested in, sort of biology and sort of life. I very much like the sort of outdoors. So I guess I was always interested in that. I didn't know if that was going to be a career or not. My mom was a housewife, and my dad was actually in the insurance business. He was an insurance adjuster that, sort of goes around and looks at sort of claims and their veracity or not. He had a very sort of inquisitive mind and was actually, quite well educated in terms of, sort of Shakespeare and the classics. And he was also a bit of a perfectionist. So I may have, sort of picked up some of those tendencies from my parents. But again, I grew up, I was an only child. So I guess my sort of early brothers and sisters were not people; they were our family dogs. So, I don't know if I sort of maybe I'm better at sort of managing canines than I am people, and so yeah, I think that and then I think when I went to sort of being an undergraduate and sort of got introduced to some of the, sort of biology and zoology, I sort of mentioned sort of the sea urchin biology and sort of watching, fertilization happening and, sort of what sea urchin, fertilized sea urchin eggs do to sort of prevent, sort of multiple fertilization events. They basically put up this barrier after the first sperm gets in, and then it actually sort of shields the other sperm from sort of delivering their payload to the egg. And then watching development, through a microscope, I thought that was just, fascinating. And but I also leaned towards chemistry as well. So I think it was, I would say that in terms of, the career-defining moment, it was sort of later in my undergraduate, degree, as I said, when I got to go to Woods Hole, to the marine biology laboratory, and spend the summer there.
So we really got to learn a lot about your academic pursuits, and there were quite a lot of them. So I'm sure balancing all that academic work and that hard work for your career might have been difficult against a personal life. And could you share insights as to how you perhaps manage this balance between your work and your personal life throughout your career? And importantly, what advice would you be able to give us young scientists to be able to better manage that aspect?
Well, I think to really sort of do science at the highest level takes, a real sort of commitment. We didn't, and I think Randy also pointed this out, we didn't really think much about, sort of life-work balances when we were, graduate students and postdocs and maybe even, sort of junior faculty. That's different now. So, I mean, I think there are, sort of generational differences. And I think, again, I what Randy said about this resonated with me, and that is that, if you want to be, a successful scientist, you have to really sort of focus and, sort of really take that endeavor, very, very seriously. And if you want to have a life outside of science, again, organization is really the key. And I mean, for me, maybe I'm just not that organized. So I would still spend, sort of most of my time, sort of in the laboratory, because that's what I like to do. Maybe playing Frisbee with the dogs, on occasion, and, sort of dinner out every once in a while or whatever. But I really sort of enjoy not only the science that I do, but also just, perusing the science that others are up to. But I think organization can allow you to sort of manage both. And even though I haven't been a great role model on that front, there certainly are, sort of many others out there, sort of like Randy. I am continually impressed with, some of our female scientists here at Rockefeller, and more generally, Jennifer Doudna, amazing, has a family, but then does everything else. We also have, sort of examples of that, sort of here at the Rockefeller. And I think, it's possible, but you probably can't be, just sort of hanging out in the lab all the time, having sort of random conversations. But I think that is important, finding a balance there, because I think spending some time in the lab when you're not just sort of doing experiments and looking at results, spending time in the lab talking to other people, about their science, about your results, using them as a sounding board, that can really, sort of enhance your research work immeasurably. And so it's probably a continual challenge. And as we would say, different strokes for different folks. I'm not sure that there's sort of one solution that works for everybody. And the relative importance of non-science activities is going to really vary a lot from person to person.
Definitely. Thank you so much for that answer. And I must say, throughout our talk, the dog lovers in the audience have had big smiles on their faces to know you also love dogs. And I think what I took away that was really brilliant was, as a researcher, as a scientist, as a student, just staying inquisitive and really looking at what other people are researching and not getting your head so stuck in what you're doing, and broadening your horizons and incorporating that into your balance of your work and home life.
Definitely.
Yeah, I mean, I think you all the time will be, sort of noticing, I'm sure that you guys are aware of, sort of how these new technologies, the automated sequencing that has really just sort of gone through the roof, with next generation methods, the functional genomics sort of technologies that are now possible to do with, sort of CRISPR screens and others, and what that can tell us about, virus-host interactions. And there are all kinds of advances that are, sort of, it's kind of like a smorgasbord of, sort of possibilities that you can think about and maybe apply to, sort of the question that you're, sort of really trying to tackle.
Thank you. So before we open up questions to the audience, we just have one last question from our side, and probably to us the most
so
important one.
Definitely. The question is, what advice would you give to aspiring researchers looking to make a significant impact in their field?
Well, I think you have to, sort of live and breathe, sort of what you're curious about. And, certainly applying, sort of your, attention to these issues and, sort of working hard and, I think responding in a positive way to sort of setbacks. At some point you have to just roll up your sleeves, and if something hasn't sort of worked as you believe it should, you have to use the resources around you to, sort of improve your chances. I think besides, sort of reading the literature, your colleagues and people are, sort of your greatest assets. And you can learn a lot from sort of other people that are, sort of successful and succeeding. And even when you're, tackling a new technique, it probably doesn't make sense to just go off and do it. But if there are other people that have successfully done it around you, sort of reach out, get some advice, sort of collect data on the best way to do something. And then you may end up, actually doing a better job of it than any of the people that you've actually gotten advice from ultimately. So anyway, those are just a few random tidbits.
I love that sentiment of learning from those around you. And just looking around the audience today, I know that there are many people who've inspired me, and I mean, just our team members within the society and people at this event, are keen to learn more and are keen to learn from each other and to learn from you. And I think that's so wonderful.
Absolutely.
So we're ready to open up questions to the audience. We might see how it's going to work with the mic and whether there'll be an echo.
So because of technical issues, you'd either have to come up here and ask your questions to Dr. Rice from the podium, or repeat your question to one of us to say to him. So your choice. Does anyone have any questions?
There's one. Would you like to come up?
This is an opportunity to come up to the podium.
I know, it's terrifying.
Pass it up.
Can you hear me?
Yes.
Just anywhere in this.
Okay. Okay. Thank you for your great talk, Doctor. My name is Taplin. I'm a postdoc at the Molecular and Cell Biology Department here. I was wondering, both you and Prof. Schekman, sorry, talking about resilience and being obsessed and, obsessed may be the wrong word, obviously, but kind of keeping going at your goal. And I was just wondering on your thoughts on that in the very fast moving science field at the moment, regards to things like big data, the publish or perish mindset, and, kind of just the collaboration, which takes a little longer but gives you better results. I was just wondering on your sort of thoughts on that and how we could perhaps balance that between, obviously we need to develop our careers, but we also want to get good science, and make a difference. Thank you.
Yeah, no, I think you, Caitlin, you bring up a really a fantastic point there. And, I think it's clear that if we just sort of take the SARS-CoV-2, the COVID-19, sort of pandemic as an example, where, as virologists and epidemiologists and, sort of public health and all that sort of thing, we were sort of thrown into the fire by the emergence of SARS-CoV-2. And we witnessed, sort of a lot of, collaborative work around the lab, sort of new ways of sort of disseminating information more quickly than the sort of usual publication route. And, I hope that is, a template for sort of moving forward in the future. But I think one of the things that you sort of bring up with this question is, sort of, if you have sort of a lot of people that are involved in this, and it's a huge sort of team effort, how do people sort of stand out from that And Randy sort of mentioned this, sort of the rewarding the individual versus the group. And I think that is something that we need to work on, because, I mean, even if it's, a relatively, sort of non-competitive area, of which there are very few these days, I think in order to produce the best science, often you have to sort of identify and collaborate with the best partners to really do that. If you're not a structural biologist, but you really need to have an atomic, sort of view of something that you're interested in, you want to align yourself with, and collaborate with the best person to sort of do that work and sort of get the job done. And that usually takes time to establish. And I mean, certainly we saw with the pandemic that there are these papers with sort of 100 co-authors, and sort of a very nice piece of work, but sort of how do you get recognized? I think we need to really do a better job when we are if you're finishing your postdoc and you publish some papers and you're sort of applying for faculty positions, we really need, in terms of evaluating and recruiting people for a postdoc position or a faculty position, we need to really look at the scientist, and we need to figure out what that person has contributed. And I think we will be relying also fairly heavily on letters of recommendation that are sort of written for people that have sort of worked closely with the applicants. But it is an issue, because I think the scientific endeavor has grown a lot, and what's expected in terms of publishing a good paper has expanded quite a lot. I mean, when I was kind of growing up as a graduate student, if it was 3 or 4 figures and sort of one, and you were contributing something new, that was fine. That was worthy of sort of a publication. But now, I mean, we get these papers that have sort of 4 figures with 25 panels each, and supplementary information that goes on and on and on. And that's a lot different. So I don't know if that answers the question, but I think there's not an easy answer. And but the most important thing is to really try and sort of look at the science and the scientists that have been involved in doing that, and sort of what they've actually contributed, and also what their promise is for the future based on your interactions with them.
Thank you so much for that answer. I think definitely a difficult question to answer, but I'm sure you covered some of the points and the directions we need to move in. So any other members of the audience with a question? Yeah, do you want to come up or?
Hello, Dr. ARS. Thank you so much for this very inspiring and interesting talk. My name is Talita Miller. I'm one of the MD PhD students here at UCT. And I was really interested when you shared about your career journey, how you had that 15 years time of really working on the research after discovering the virus to even when the life cycle of the virus could be done in cell culture. I'm sure that you must have had many dead ends along the way. And I think, especially as a young researcher, one of my questions to you would be, how did you know when to stop pursuing an idea that ended up being not fruitful? And when did you know to persevere in an idea that did end up working?
Yeah, I guess the challenge there is, sort of when you have an idea and you're exploring it, is to sort of decide, is there an experiment that you can do that will tell you whether or not this is going to be, a productive, sort of research direction to pursue. And I guess that's the go, no-go experiment. Or is there an experiment that I can do that is going to say, this just wasn't a great idea. So I think that's important. I think in my sort of personal case, which was really trying to, create a robust genetic system that we could use to study hepatitis C. I just, it was an interesting transition because I'd been working, as I mentioned, on yellow fever. And, even when I would go home and my mom would ask me, what are you studying these days? And I'd say, well, I'm working on yellow fever. And she'd go, well, don't we have a vaccine for that? Why are you working on that? Which was true. But by, sort of working on that virus, we learned a lot, which when hepatitis C sort of was discovered, we could apply to studying that virus. And I think one of the things that helped me sort of get over the successive roadblocks that we faced in studying hepatitis C was the sort of struggle and the successes that we made on yellow fever to, sort of create a genetic system for studying that virus, to sort of struggle with, sort of finding the complete, sort of RNA viral genome sequence. I really felt with hepatitis C that it should be possible. The other thing was really working on a more sort of fundamental model system, as a molecular virologist, and then, basically have this virus that had a huge, sort of clinical importance, and, sort of not only being thrown into the challenge of sort of working on something that we could not get to replicate in the laboratory for many, many years, but also the realization that there were, potentially hundreds of millions of chronically infected people out there with this time bomb that was cooking. And as a basic scientist, getting to interact with, clinicians that were trying to sort of deal with that patient population with very limited ability to really treat them effectively, and also interactions with the patients themselves. Even when we would make, sort of some of these, and others would make fundamental breakthroughs, you end up, having patients or the parents of children that were chronically infected with hepatitis C contact you and ask you, sort of what's going on, sort of when is a better treatment going to be available. So all of those things, I think, really helped in the case of the hepatitis C work to sort of keep doing it despite the setbacks.
I think that's really amazing, and I think sometimes when the value of your research and the value of your question is so important, you kind of pester on and push on no matter what stands in your way to really try and answer that question. Is there anyone else in the audience who'd like to?
Hi, Dr. Rice. My name is Max Mendelo. I really appreciated your talk. I was just hoping to get a comment on a phrase I've been seen making the rounds lately, and that phrase is the democratization of virology, which basically speaks about we are one or two breakthroughs away from the powers of virology being in the hands of a much larger pool of people, for better or for worse. And I was wondering what you think about that.
Well, I don't know. I mean, I guess I might like to sort of have you clarify the question. You can head back up there. I mean, I guess, I think that in terms of advances in virology, I guess, are you worried about sort of the good, the bad, and the ugly, or what's the underlying reason for your question?
So the good and the bad and the ugly is a good place to start, but it's when it's accessible to the average layperson, much higher levels of access to virology, whether that be research or manipulation or any of the dangers that you see your field potentially holding to anyone, as well as the benefits.
Yeah, I mean, I think there are always sort of a good side and a bad side. I mean, I do think that the public now realizes, sort of what a new virus, sort of in an immune naive population that can spread efficiently can do. And, this has, sort of engendered, sort of a lot of, discussion about, okay, sort of where did the virus originate? Where did it come from? Sort of what sort of nefarious things could, sort of be at play here, which I don't really think is a likely scenario, although these things are hard to disprove with 100% certainty. And there is a lot of new power, sort of in the biological sciences, in the tools that we actually have to explore. And there is a lot of sort of attention being made to the so-called sort of gain of function experiments, where scientists are trying to understand if you, for instance, have a drug that's successful at treating sort of a viral infection. One of the things that you'd like to know about a drug or a compound under development is, can the virus, sort of gain resistance to that compound? And is the sort of resistant virus, sort of still capable of efficient replication and spread and things like that? I mean, we've certainly seen that, sort of in the case of monoclonal antibody, for example, resistant variants of SARS-CoV-2. And I think it's very important to understand that if you really want to come up with, sort of longer lasting treatments or alternative treatments where, the virus can get away. But there is a lot of discussion about, these kinds of experiments and the kinds of transparency and disclosure that needs to take place in order to conduct them sort of safely and get the most out of that kind of work. So, I mean, I think we're seeing that too, not only with virology, but with the ability to do gene editing, at a specific locus in the human genome. And, sort of what does that mean in terms of treatments or, sort of engineering? So I think it's, these are discussions that are important to have as a society.
Definitely, and thank you so much for unpacking that, Dr. Rice. Definitely very important discourse that needs to be had continuously as the scientific community. But right now in the audience, does anyone have one last question before we close? Yes, do come.
Hello, Dr. Rice. My name is Fee Win. I'm a second-year medical student at the University of Cape Town. Before I get to my question, I just want to personally thank you for giving us the opportunity to speak with you and overall your contribution to physiology and medicine and the whole world, really, and giving us the opportunity and many other young scientists and scientists abroad to build up on our current knowledge.
Well, I expect you guys to grab the torch and carry it going forward.
So my question is, since winning the Nobel Prize, do you now feel pressured, or do you feel under pressure to produce more quality science? Or after winning the Nobel Prize, has it kind of emboldened you to pursue other questions?
Well, I don't know. I've actually been pretty lucky in the sense that for very much the majority of my career, I've been able to sort of do what I was interested in rather than, sort of what other people thought was interesting. And, occasionally those have converged. I do think, and I don't know if you saw this sort of recent perspective in Science magazine, that, sort of when you sort of win the Nobel Prize, it's not necessarily the best thing for your scientific career. and because sort of you have other demands on your time, and I think it's important to take some of these opportunities like this one to sort of communicate with sort of the next generation of young scientists, because I think this is an enterprise which is very important locally and globally. So I think that at least for me, even though you guys would say I'm old and I should be retired anyway, I still have a fairly active sort of laboratory with about sort of 30 people that are doing various projects in the lab, and you have to sort of make sure that they have the resources that they need to do what they want to do, and so we are actually having some after spending nearly 30 years working on hepatitis C, we're returning to some of our studies on sort of mosquito-borne and tick-borne sort of viruses, and other studies as well. I mean, obviously, we're continuing to do some work on coronaviruses and other viruses, and we are branching out a little bit, but that's driven to some extent, depending upon what I'm interested in, but also what the people that are coming to the lab are interested in. And I see my role these days to really sort of provide an environment where people can come and test their ideas and maybe do something quite adventurous that would be more difficult to do in other environments. So it's kind of a mixed bag. I think in terms of having time to spend with your own research, the Nobel Prize is probably not good because you've got less time. But in terms of sort of being able to sort of talk to and sort of motivate young scientists, it's a nice thing to be able to do, and it does give you sort of that opportunity. So yeah, I have to say, sort of since we're coming to the end here, that I was never doing what I did, and my colleagues were not thinking we're doing this stuff to win prizes. I mean, you do it because you have a passion for what you're doing, and then sometimes these things just happen. And that may be a little bit different than some of the other laureates, but I think that was also true of my laureates, Harvey Alter and Michael Houghton. Sort of Harvey noticing something that was going on in his clinical practice and getting very interested in it. What's causing this? And Michael Houghton and his group at Chiron really sort of going after to try and sort of clone this entity using some very cool tricks at the time. So it's really all of these things are just an incredible, group effort, and certainly hepatitis C is no exception, with really thousands of people involved in this success story.
Thank you so much for that response and for really highlighting the key aspect of passion in any area of research. And thank you for answering all of our questions and for being part of this discussion with us tonight. And we will now be moving on to the vote of thanks, which will be given by Professor Reddy, who is a South African scientist, an emeritus professor, and our very own interim vice-chancellor at the University of Cape Town. Professor Reddy holds the South African Research Chair in Computational and Applied Mechanics and is the director of the Center for Research in Computational and Applied Mechanics there. So please give us a warm welcome for Professor Reddy.
Well, good evening, everybody. Thank you for that introduction. Gosh, what a wonderful evening this has been, and what a pleasure it is for me to be able to, say a few words along the way to delivering a vote of thanks. So, what is it about research? What is it that drives us? We've heard a fair bit from our two esteemed Nobel laureates about exactly that. We've heard terms such as curiosity, the drive to understand, and we've also heard about obsession, a real obsession with wanting to know something, with wanting to figure something out. It was said by somebody, actually a vice chancellor at another university in another country, that research is an expression of faith in the possibility of progress. There is implicit in what we do in doing research that we are able to make progress, and the research that we go about doing is an expression of exactly that sort of faith. So, as you know, I'm sure UCT takes research very seriously. We sometimes describe ourselves as a research-led university, at other times as a research-intensive institution. I'm not sure about where the boundary lies. I think that is less important than being aware of the fact and embracing the fact that we take research very seriously. It is a core part of what we do within broader context of scholarship, whether that's teaching and learning and other forms of scholarship. And so we recognize the centrality of research in the scholarship, which brings me to the African Research Society, which I have to tell you I learned about for the first time not that long ago. And I'm very, very privileged to have learned about the society and to be with you here tonight. I wanted to comment on one aspect of the goals of the society, which has to do with Africa-centric research and researchers in Africa. And this evening's event, the interviews, the discussion, the questions and so on, really brought me to ask the question in relation to the objectives of the society. Well, what is it that we must do to ensure that we, and when I say we, I mean all of us in Africa, that we are part of it all, that we are not simply recipients and users, but also contributors and part of that network. As somebody once put it, don't just invite somebody to the party, ask them to dance. And then somebody else said, well, and also invite them also to choose the music. So really being part of it all. And I think that the work of the African Research Society is so important in this respect. And I'm hearing you're second year undergraduates, we are not, as far as I know here, postgraduate students, postdocs and the like, but you are at the beginning of your university careers and you have this intense interest in research, and that is so inspiring and that is so encouraging, and that is what will help us to achieve the goals as set out by the African Research Society. I would like just to say a few words about what has emerged from these fascinating discussions and presentations that we've had this evening. So this is somewhat selective, but we've heard about the accidental nature of choosing one's career, the accidental nature of research, serendipity, if you like. But I have to say, it's something that used to concern me in the sense that I would encounter something which really led me to make real progress with whatever problem I was working on. And I'd worry about the fact that, if I hadn't done this, if I hadn't opened that book, if I hadn't spoken to this person, I would never have gotten here. And, oh my goodness, now, is this a problem? It's not a problem. It's very much part and parcel of it all, as we have heard. Being open to accidental changes in direction, to serendipity, and to being inspiring in that way. The second point that I'd like to highlight concerns certainly what Professor Andy Schekman had mentioned about confidence, being confident about what you do, approaching it with confidence, being willing to take risks. And there's a difference, I think, between taking risks and being reckless, calculated risks. Peter Medawar, who was a very famous British scientist, biologist, wrote a wonderful book called Advice to a Young Scientist. And in the book, he indicated that one of the attributes of a good scientist is what he called a sanguine attitude, which is really saying the same thing. You have to have a positive attitude to your work, notwithstanding the knocks, and there are most certainly knocks that come along the way. But you would not succeed if you went through it all, with your head down and dwelling on the failures. So it needs that kind of confidence and, of course, focus as well. We've also learned or heard about taking complicated problems and breaking them down. I think this was in response to a question about how do you go about things when you are dealing with a problem that seems to be intractable. In my area of mathematics, there was a very well-known mathematician, Hungarian mathematician called George Polya, who put exactly this point in a similar way by saying that if there's a big problem that you can't solve, then there's a smaller problem that you can't solve. In other words, you break it down until you get it down. At least in mathematics that works a lot of the time, until you have something that you can make some progress with, and then you make your way back. And importantly, we've heard quite a bit about collaboration, the importance of collaborations, networks in research work, but I think also what I've learned from what we've heard this evening, the human side of it, what really makes our work and our lives worthwhile, the progress that you make in research, but also the fact that one develops these wonderful relationships with colleagues at your own institution and around the world, and of course with younger colleagues, with your proteges, if one is a mentor, and on the other side with your mentors. So that is such a valuable part of all of it. So those were just a few of the points that have come to the fore in my mind in the course of this evening. Let me, however, turn to extending my thanks, and I begin by saying what a real privilege it has been to have had with us this evening Professor Charles Rice and Professor Andy Schekman. Thank you for your generosity, for your willingness to share a fair chunk of your time with us. It has been so inspiring. We have learned such a lot. We know, as somebody has indicated, what the demands would be like on your time and on your lives as Nobel Laureates. So we are very deeply appreciative of your willingness to spend time with us and to allow us to learn so much about yourselves and your work. Then I'd like to thank our keynote speaker, Professor Salome Maswime. It's always a privilege to hear you, Salome. You have such an infectious enthusiasm for your work and for scholarship and everything we do, so thank you to you. And then let me thank our office bearers and members of the African Research Society, and Elisa and Wahid, who welcomed us, and then Teo and Sayurin, who had a difficult task, but which you acquitted yourselves with such confidence and poise, and I thought, by the way, that the questions that we had from the audience were wonderful questions. Thank you very much. And thank you all for being here. It really has been a privilege to be with you this evening. Thank you.
power of genuine curiosity and how this is the driving force behind science. We've heard of how hard work and determination are crucial to becoming a good scientist. And above all, we've heard that there's no way of sitting down to make your scheme to win the Nobel Prize. It is simply a product of continued enthusiasm for your area of research. Something that I took away was the need for us to build systems that are more team-oriented. At the moment, we live in a world that favors the individual, and it's crucial for the success of all research endeavors going forward to cultivate teamwork and collaboration. Running events like this takes a lot of effort, and most importantly, a lot of money. And the main reason I've come up here today as the treasurer of the African Research Society is to appeal for more money to keep doing nice things. Aside from hosting our second annual Inside a Researcher's Mindset event, we spent the year getting up to a lot of things. We host routine mixes to bridge the gap between science departments and the students they teach. We have organized our first intake of the Interdisciplinary Research Apprenticeship Program to cultivate the next generation of South African clinician scientists. And we've also hosted our Order of Mapungubo event, which commemorated South African science legends. All of these events have brought our operating costs to an excess of 50,000 Rand for this year alone. We already have plans for a bigger and better IRM next year, which may or may not include a new, bigger venue of national or international importance. And similar to an exciting new match on a dating app, we want to take this off the app and into real life. I've said what I need to say, get from that what you will. But to make a contribution, please scan the QR code that is shown on the slide. Alternatively, I believe there will be a card machine going around, which directly into our fund. Guys, I'm serious, no amount is too small. Unless it's less than 2 Rand, then it's too small and the machine won't accept it. So think of this as you purchased your KFC meal, now you're adding 2 Rand.
So I'm Robin Beach. I think my picture, oh there it is. So we're back to that. I must say, my blood has rushed to my legs, so bear with me. Esteemed guests, what an incredible evening it has been. As we come to the close of this remarkable event, I am filled with a deep sense of gratitude and inspiration. We have been privileged to witness the conversion of dedication and brilliance as we gathered here under the banner of the African Research Society. Our commitment to African-centric knowledge, progress, and collaboration has shone brightly today, and I want to thank each and every one of you for being a part of this remarkable occasion. The presence of esteemed individuals such as Prof. Solomon Maswime and Prof. Diane Reddy, as well as our distinguished guests, Nobel Laureates Prof. Randy Schekman and Dr. Charles Rice, underscores the significance of the work we do and the potential impact we can collectively achieve. Their insights and expertise have not only enriched our understanding, but have also kindled flames of curiosity within us. Throughout the course of the evening, we have engaged in thought-provoking discussions, shared innovative ideas, and witnessed the power of collaboration in Africa and beyond. Our sponsors have played an instrumental role in making this event possible, and we extend our deepest appreciation to them for their unwavering support. I'd like to note a few takeaways that I got from this event. Dr. Charles Rice mentioned, you don't plan to win a Nobel Prize. In the pursuit of scientific discovery, the aspiration for recognition is often secondary to the dedication of expanding our understanding of the world. True breakthroughs emerge from a genuine passion for uncovering the unknown, and this intrinsic motivation fuels the profound discoveries. By focusing on the process rather than the goal, we create an environment where innovation can flourish naturally, paving the way for unexpected and impactful contributions to science and society. He also mentioned that fundamental researchers, when they see their research implemented in the clinical platform and saw it has a positive impact on them. And I think the bridge between fundamental research and clinical application is where the true potential of scientific progress is realized. The role of the clinician scientist is not confined to labs or hospitals alone, and Prof. Maswime is a big champion of this. It's about translating research, theoretical insights, into tangible benefits for patients. The integration of research and practice exemplifies the synergy between scientific exploration and compassionate patient care, underlining the essence of bridging disciplines for the betterment of human health and well-being. Dr. Schekman mentioned the profound impact of insulin. One of those poignant examples of research impacting life-changing, translating into life-changing impact lies in the discovery of insulin. The journey from fundamental research to clinical implementation is exemplified by the story of insulin, which revolutionized the treatment of diabetes. This breakthrough underscores the transformative potential of collaborative efforts between scientists and clinicians, and by understanding the intricate biology behind production and function, researchers laid the foundation for a treatment that has not only extended countless lives, but also symbolizes the remarkable possibilities that emerge when research meets real-world application. As we reflect on the wisdom shared by Prof. Randy Schekman and Dr. Charles Rice, I'm reminded of a quote by the renowned Isaac Newton. If I have seen further, it is by standing on the shoulders of giants. And tonight, that is exactly what we've done. And now it is our responsibility, as Prof. Diareti said, to carry the torch forward, to inspire the next generation of researchers, innovators, and leaders. The African Research Society was born of a collection of visions, a collective vision for progress, a vision that transcends boundaries, embraces diversity, and empowers every individual to contribute to the betterment of society. We are not merely witnesses of history, we are the architects of it. Let us harness the knowledge we gain, the connections we forge, and the passion to drive us to create a brighter future for Africa. As we conclude this evening, I encourage each and every one of you to continue fostering meaningful collaborations, to push the boundaries of knowledge, and to remain steadfast in your knowledge, in your commitment to excellence. Let's remember that our pursuit of knowledge, of understanding, is a journey without end. And it is in our united efforts that we will shape a legacy that resonates through generations to come. Thank you for your participation, your dedication, and your unwavering belief in the power of research. Let's go forth from here with renewed determination and a shared sense of purpose. Once more, if you're able, please consider making a donation. Your generosity is truly appreciated. QR codes are conveniently available for your contribution. Or again, I will be carrying the Yoko machine around my neck, so you just wave to me. After these enlightening discussions, indulge in the array of food and drinks just outside the lecture venue. Lastly, to help us minimize waste, kindly drop off your name tags at the designated table downstairs. And good night. May our collective endeavors continue to illuminate the path.